Welcome to our class, Medical Mycology. My name is Mr. Kingsley, and I'm going to be taking this course. So the word myco can mean fungi or myx, which is mushroom. Logy is the study of. So mycology is simply the study of fungi, and this will include their genetic, their biochemical properties, their taxonomy, and their uses to human as source for medicine. For example, our common penicillin. Foods like beer, wine, cheese, and mushrooms, as well as the dangers these fungi pose, such as poisoning or infections. Now, medical mycology, on the other hand, is a special uh, branch of the medical microbiology, which is concerned with all aspects of diseases in human as well as in lower animal that are caused by pathogenic fungi. Mycosis are diseases of humans and lower animals that are caused by pathogenic fungi. So mycosis is the diseases. Medical mycology is a special branch of medical microbiology that deals with the formation of disease by fungi. Now, let's kind of start by looking at definition of fungi. Fungi are eukaryotic cells that contain membrane-bound cell organelles, which include nuclei, the mitochondria, the Golgi apparatus, the endoplasmic reticulum, the lysosome, and others. They are also heterotrophic, meaning that they lack chlorophyll. And because they lack chlorophyll, they cannot carry out photosynthesis. So they are not autotrophic, just like plants and algae. Rather, they are heterotrophic absorptive organisms that are, many of them are saprophytes, as they like to live on dead organic matters, while few of them are parasites. These utilize living tissues. Then fungi, like plants, have rigid cell walls. They are non-motile as well. And this is a characteristic that separate them from animals. Now let's look at the structure of fungi. Fungi occur in two basic growth forms or stages. These stages are one, what we refer to as a unicellular or the yeast form, and B, a filamentous or mold form. Now let's look at the yeast form. The yeast form is defined morphologically as a single-celled fungus that will reproduce by carrying out budding to form what we call blastoconidia, the form we call blastoclonidia. As you can see, now this is the parent cells undergoing budding to form the new blastocodia. This is a blastocodia. So this is formed. You can also see pseudohyphae shooting out here. So this is an illustration of how the fungi produce. So this is a yeast form, illustration of the blastoconidia that you find in figure 1. Now, a filamentous or mold form is a vegetative growth of filaments, which consists simply of number of filaments that are packed together. And reproduction is by spore, or what we call conidia. Spore or conidia. So fungi filaments are known as the hyphae. This is the hyphae, as you can see here. Hyphae is growing. And the mass of hyphae collectively will make up what we call the mycelium. So when all these comes together, they give us the mycelium. Now, there are two kinds of hyphae. There's what we call the non-septates. This is a non-septate hyphae, and the septate. The non-septate can also be known as the sinusitic hyphae, while the septate are the ones that are segmented. Now, apart from the fungi, there are also what we call the dimorphic fungi. Now, this class of fungi are kind of different from the yeast of the filamentous ones. This class of fungi is characterized by two forms of growth. Now, the first one is that they grow as a mold with septate hyphae in their natural reservoir, that's in the soil. When they are incubated at 25 degrees centigrade, that's the room temperature, on conventional fungal media. Most of the fungal media, we can use sabroid dextrose or potato dextrose agar. So these ones will grow. Then they also grow as yeast in the tissue of an animal or a person when they are incubated at the temperature of human at 7 degrees centigrade on enriched media, the brand heart infusion agar. Now, antifungal agents. An antifungal agent is a drug that selectively will eliminate fungal pathogens from a host with minimal toxicity to the host. So what we're saying is that these fungi drugs will eliminate the fungi, but will have no effect on the host. Now, there are different fungi, antifungal drugs. Like we have number one, Pauline antifungal drugs. They include amphotericin, nystatin, pimarisine, which interact with steroid in the cell membrane of the fungi, or ergosterol in fungi, while cholesterol in humans. Those are steroids to form channels through which small molecules will now link from the inside of the fungal cells to the outside, causing the death of the fungi. So that's how the Pauline antifungal drugs react. This amphotericin or nystatin, they help to destroy the membrane of the fungi. We also have the axo antifungal drugs like your fluconazole, itraconazole, or ketoconazole, which inhibits cytochrome P450 dependent enzymes. Now, this one is involved in the biosynthesis of ergosterol, which is for the formation of the membrane, which is required for fungal cell membrane structure and function. So antifungal axos are synthetic drugs. So these ones will also help to destroy the formation of the membrane. So when there is no membrane formation, it leads to the death of the fungi. Another antifungal agent is allelamine or morpholine antifungal drugs. Allelamines, we include nifedipine, terbinafine, which also inhibits ergosterol biosynthesis at the level of squalene epoxidase. The morpholine drugs, amorphine, will inhibit the same pathway just like allelamines. Then we have antimetabolites, antifungal drugs, such as 5-fluorocytosine, which acts as an inhibitor of both the DNA and RNA synthesis via the intracytoplasmic conversion of 5-fluorocytosine to 5-fluorouracil. So these ones will usually affect the formation of RNA formation in the fungi. Now, how do we diagnose mycosis? So let's look at lab diagnosis of mycosis. Now, there are different ways to carry out diagnosis of mycosis, that disease of fungi. We can have clinical presentation. First, we look at the history, look at the risk factors, then we look at physical exam, that look at lesions, pick some lesions. Now, that will tell us whether it is mold or yeast, whether they are septate hyphae or not. These are things we look at clinically. Then, of course, we can culture the organisms by days to weeks. Now, most media that are used for the quantification of fungi, like we mentioned, are sabroid dextrose agar SDA or cybroid dextrose agar with antibiotics in order to remove contamination. We can also use brain heart infusion agent or problem contaminating bacteria, we use brain heart infusion agent. Then we can also carry out serology or antibody or antigen tests, and also use molecular biology, such as PCR. You can use PCR to also conduct it. Now, classification of diseases that are caused by fungi. What this says that mycosis is a disease that are caused by fungi. Now, these can be classified into 5 based on the levels that they initially colonized. According to the level initially colonized, we have superficial mycosis, we have continuous mycosis, we have subcutaneous mycosis, we have systemic mycosis, which is due to primary pathogens. We also have systemic mycosis due to opportunistic pathogens. Let's take them one after the other. Let's start with superficial mycosis. These are superficial fungal infection of the skin or hair shaft. So it doesn't invade the living tissues. So no living tissues is invaded and there's no cellular response from the host. So essentially, there's no pathological changes that are elicited during this. And they can be Malaysia infection, tinea nigra, one that infects the nails, black pedra, white pedra. These are superficial mycosis. Let's take them one after the other. Let's look at Malassezia infection. Now, Malassezia infections are what we call Malasseious or pityrosporosis. They are common conditions without serious nature. They are usually due to commensal yeast known as Malassezia genus. These Malassezia yeasts are known long before in the human pathology. The best known is the Malassezia fufu. Now, let's look at the epidemiology of Malassezia infection. Let's talk about the skin commensal yeast. Malassezia will proliferate in the epidermis, producing mycelium under the influence of various factors that are specific to the host. One of the factors is that the place is oily. So high content of triglyceride and free fatty acids, or the application of fatty substances on the skin, like solar oil. Then heat, humidity, sweating also encourage these Malassezia fufu. Pregnancy is another condition that can allow them because it leads to immunosuppression. So there's probably a genetic predisposition. One thing we are sure of that the Malasseias are not contagious. Now, clinical manifestation is that they have what we call pityriasis vesicular. Now, this is a chronic superficial fungal disease of the skin. It is characterized by white, pink, or brownish lesion. You see it's there. You see colors, often covered with thin phosphorescence scale. The color usually varies according to the normal pigmentation of the patient. So if the patient is black, you see it's black. If it's brown, you see it's brown. So exposure of the area to sunlight as well as the severity of the disease, the term vesicular, which may colors. So that's one of the Malassezia fufuri. We also have the seborrheic dermatitis. Now, the seborrheic dermatitis is a common, harmless, itchy, red skin and scaling rash affecting the face, the scalp, and other areas. We can have pityriasis capitis, that's dandruff. This is the dandruff of the scalp. It's usually favored by stress. Seborrheic itching is common and can also cause hair loss. Malassezia folliculitis is a skin disease, especially that is prevalent in young men. Itching is normal. The usual seat is the back, with a possible association of the front of the chest. We also have the Fujimiya malassezia. These ones are rare and they usually occur in premature infants or immunocompressed fed by intravenously. Now, how do we diagnose these? Laboratory diagnosis will involve one clinical material. The skin scraping will scrap from the patients with superficial lesion. You can use bloods and indwelling catheter tips for patients with suspected Fujimia. We can also use direct microscopy. Skin will be molds in 10% potassium hydroxide, Lacero, and Peka ink solution. It will show a characteristic cluster of thick wall, round body yeast-like cells and short angular hyphae form up to 8 micrometer in diameter. As we can see in our diagram, all this was you see. This is what we're seeing in a petri dish. So they indicate the presence of these Malassezia fungi. Now, these fungi can be cultured in vitro. So in vitro growths must be stimulated by natural oil or other fatty substances. The most common method used is to overlay sabroid dextrose agar containing actidoin with olive oil. Let's look at the serology. Of course, there are no currently available serological procedures for the diagnosis of Malassezia infections. Treatment, we usually use a topical imidazole in a solution, or we use ketoconazole cream, which has proved to be very shampoo to wash the scalp. In severe case with excessive lesion, or in cases with lesions that are resistant to treatment, oral therapy with either itaconazole will be very effective. The second one is Tinea nigra. The etiological agent is Exophilia warnecki. Now, the skin manifestation, the skin lesions are characterized by brown to black macules, which usually occur on the palmar aspect of the hand and occasionally the plantar and other surface of the skin. The lesions are non-inflammatory and non-scaling. Laboratory diagnosis, these are skin scraping. You can also use direct microscopy. Skin scraping should be examined using 10% potassium hydroxide and Packer ink or colorful white mounts. You can culture them using primary isolation media like the cybrodextrose agar. Serology is not required for diagnosis. Treatment, we use white feed ointment or topical imidazole in a solution. Another fungi of interest is the black pedra. The black pedra is a superficial fungal infection of the air shafts, which is caused by Pedra hortae, which is an ascomycetus. The clinical manifestation is that infections are usually localized to the scalp, but may also be seen on hairs of the beard, the mustache, the pubic hair. The black pedra mostly affects young adults, as well as epidemics in families have been reported following the sharing of combs, hair brushes. Infected hair generally have a number of hard black nodules on their shafts. So the laboratory diagnosis is good epilepted hair with hard black nodules which are present on the shafts. We can also use direct microscopy. Hair should be examined using 10% potassium hydroxide. Nodules are made up of a mass of pigmented with a stroma-like central containing asc. So how do we culture? Hair fragments should be implanted onto the sabroid dextrose agar. Then you'll see colonies of the Pedra hortae, which are black or brown black, and it will take about 2 to 3 days or weeks to appear. Serology, not needed. So the white pedra is a superficial cosmetic fungal infection of the air shaft, which is caused by Trichospora species. Now, the clinical manifestation, infections are usually localized to the axilla or scalp, but may also be seen on facial hairs and sometimes pubic hair. The white pedra is common in young adults. The presence of irregular, soft, white or light brown nodules, uh, which are 1 to 1.5 millimeter in length, and they are firmly adhering to the hair, is the characteristics of the white pedra. The laboratory diagnosis, epilated hair with white soft nodulated present on the shaft. The hair should be examined using 10% potassium hydroxide as well as Packer ink or Cacoflow white mounts. Look for irregular, soft or white or light brown nodules. Hair fragments will be implanted on the primary isolation media like the sabrod dextrose agar for culture. Serology is not needed. Treatment for black and white pedra, the hair should be shaved, or we can use ointment from imidazole seronium sulfides to actually cure these particular. So we've seen some of these fungi and how they cause problems to humans. They affect us in one way or the other. So it is very important that we get to understand most of these fungi, know how they affect us, know the benefit, the aspects, and how to take care of them. In so doing, we'll be able to combat these fungi. We've learned that mycosis and see what we can do. We cannot cover everything, but in the next class, we'll look at some other fungi of interest. Thank you.